Good evening, everyone. Welcome to our event this evening, and thank you very much for joining us. We hope that you have enjoyed the Cambridge Festival events so far and that you'll enjoy spending time with us tonight. We're going to start our event this evening with a discussion about the tactic R trial and about the local research that we've been doing on COVID-19. We invite you to submit your questions to us. And there will also be a live Q&A part at the end. So do submit your questions using the Q&A function at the bottom of your screen as we go along, and I'll be able to take those at the end. Joining me this evening, I have three distinguished researchers, all of whom work both in clinical care and research, and who have played active roles in our local research effort throughout the pandemic. I am joined tonight by Professor John Bradley, by Professor Ian Wilkinson, and by Dr. Frances Hall. Professor Bradley is the Director of Research at Cambridge University Hospitals. Professor Wilkinson is the Director of the Cambridge Clinical Trials Unit, and Dr. Frances Hall is the Chief Investigator of the Tactic R trial that we're going to be discussing this evening. I am going to open our questions this evening with a question for you, Professor Bradley. As Director of Research at Cambridge University Hospitals, you would have been involved in a lot of the preparations that we made when we were leading up to the first COVID patient being admitted at about this time last year. Can you tell us what was going on at about that time and what preparations were we making to be able to support the research effort?
You have to remember the uncertainty, actually the fear and the courage of everybody, patients, health professionals and researchers at the time. We were really very unclear what lay ahead. And the first thing that we, well, in fact, there were two parallel processes we had to undertake. One was think about the existing research on the campus and how safe it was for the participants. And we currently or normally would have about 1,000 studies running across the Cambridge Biomedical Research Campus. And we had to pause many of those. That means notifying the patients and notifying the regulatory authorities. And obviously thinking particularly around clinical trials where people were receiving drugs about the relative safety of pausing the study or continuing. Then in parallel with that, we were thinking, how can we, through our research effort, help the COVID research? The early work focused an awful lot on diagnostics. Mm-hmm. People were arriving in hospital. We needed to know very rapidly, if we could, whether they were infectious or not. And one of the earliest studies was around using a point of care test. In fact, we were the first to report on the use of point of care testing to diagnose people within 90 minutes of undertaking a test so that we could get them to the right environment to protect them and others. We were also looking at the diagnostics, particularly even at the early stages. We were aware that there were variants emerging. We weren't so worried at that stage about whether they were susceptible to treatments or vaccines, because we didn't have those. What we were more worried about was whether the diagnostic tests would still detect them. So we were doing those sort of studies in parallel. And then we wanted to try and better understand the disease. And there we exploited a resource or infrastructure that Cambridge leads called the bioresource, the NIHR bioresource. This is a vehicle for people to become involved in research. It's a national resource. There's around 200,000 people across the country involved in it. And essentially what they do is tell them a little bit about themselves, allow us to have a look at their medical records and collect a sample from which we can extract DNA and learn about their genetics. And then if we have a research study that we think they might be interested in, contact them about it. What we did was to establish the COVID bioresource. And that allowed both patients and staff who were interested in participating in research to join the COVID bioresource. And that way we could learn about how they responded to the disease and about the actual mechanisms of the disease. They were the early studies, but of course, while all that was going on, we were also setting up to run clinical trials.
Fantastic, thank you. Professor Wilkinson, you're the director of what we call the Cambridge Clinical Trials Unit here in Cambridge. Can you tell us briefly, first of all, what does a clinical trials unit usually do, and how was it different during this period of setup of lots of different studies?
Thanks. So what we normally do is help people develop new treatments to improve patients' outcome, be it drug or device, etc. So our role in that process is to work with people who have ideas and help them realize that idea, and to put together a protocol, which is a set of instructions about how the trial is going to run, to help them get the permissions which they need to run the trial and navigate that, to help input into design, statistics, collecting the data and then analyzing it. So that's our sort of normal job. And we would normally run about 40 to 50 trials at any one time and be closing some of those down and setting some of them up. And they might just involve Cambridge, or they might be several sites in the UK, or they could be international sites. So we do all sorts of trials, but we're particularly keen on running early phase trials and developing really novel interventions or repurposing drugs, which you mentioned earlier on. So as Professor Badley said, when we were all concerned about the pandemic, two things happened really. The trials unit took a decision that it wasn't going to open any new trials that were not related to COVID. And we worked with investigators to determine whether they should be closing down or pausing ongoing trials. Initially, there was a different type of response from different investigators. So some investigators just stopped everything. Some wanted to try and continue. But as the pandemic progressed, virtually everything stopped that wasn't critical. There were some critical cancer trials that had to continue. And then we set up a number of trials designed to look at COVID, including Tacticar with Francis Hall. And a number of things changed. So a lot of our staff are working from home. Mm-hmm. And we embraced something called Zoom that I can't say I was familiar with before we started. We brought critical staff back because sometimes that's the only way to get things done. And we prioritized our effort against COVID. And whatever people were working on, they started working on the COVID studies. And the regulators had changed the framework that we had to work in. So they're the people who give you ethical permission, or the government agencies that allow you to do a study with a drug. And they were working to much tighter timelines. So gone were the 30 days for a response, you probably got a phone call the next day. And we went from setting up trials in what would be 3 to 6 months to setting up trials within a month, etc. So it was a Herculean effort by all the staff in the trials unit.
Amazing, thank you. Dr. Hall, in your usual role, you work with patients that with what we call rheumatoid conditions. On the University Hospital website, you're listed as the connective tissue disease lead. I think many of the people who are joining us tonight, and many of the public, think of COVID as a condition of the lungs and the respiratory condition, and the respiratory system, sorry. So where does your expertise come in here?
Yes, I realise it's not an obvious connection to a rheumatologist, but what I normally do is look after people with multi-system autoimmune disease. So these are diseases where the immune system does something it's not supposed to do. So what it's supposed to do is attack bugs, like viruses, obviously, and bacteria and fungi. What it sometimes does, unfortunately, is makes the wrong call and attacks bits of yourself, bits of people. And then people get sick as a result of that. And so that's my day job, really, is working out how to best control these conditions where the immune system goes a bit off piste and starts attacking self. So it wasn't obvious to me at the outset of this that COVID was going to be a disease I would have a role in researching. But I started reading about the implications of COVID for my patients, thinking about my patients taking a lot of medicines which damp down their immune response, and thinking initially that this would probably be a bad thing because it might, make them more susceptible to getting a bad dose of COVID. And then as we really started looking at the data coming from China and coming from Italy, it became apparent that it wasn't really quite following that model. So although most people get COVID as a viral infection and handle it as a pretty sort of standard viral infection, they get on well and then their immune system attacks the virus and then they get better. As we all know now, a minority of people go on and get very sick. And unfortunately, some of them die as a result of it. But what became clear was that people were getting sick at the point where their learned immune response to the virus was kicking in. So this is the time when we expect them to start being protected. But a minority of people started to get even more sick at this point. And as data flowed more and more, it became clear that we were dealing with a condition that was predominantly due to the immune system causing damage rather than the virus causing damage. So this started to look a lot more like my day job. And really, that's how I got involved.
Fantastic, thank you. And after you had the idea for the trial, how was the Tactic R trial team put together to run and deliver the trial?
Well, when I started thinking about COVID as an immune-mediated condition, I started thinking, well, we should really be looking at damping down the immune response. And the majority of the focus at that stage, of course, it's changed a lot over the last year, but the majority of focus at that stage on the research front was to think of it as a viral infection and to think about tackling it in that sort of standard way. So I went to Ian Wilkinson and Joseph Cherian in the clinical trials unit and said, well, look, what about doing this? And they immediately understood the importance of the question and why that was a sensible approach. And of course, we then talked to John Bradley, and the Biomedical Research Centre infrastructure came in behind. But then we also engaged with a lot of other clinicians up and down the country who have expertise in dealing with immune-mediated diseases. So many of them rheumatologists. And many of those were supportive, and many of those people became our investigators or our clinical trial leads in the hospitals up and down the country. And two centres particularly became very heavily involved with the design and running of the study. So James Galloway at King's College London and Andy Cope at Guy's St Thomas's. And together with us, they formed the sort of key consortium. So the clinical trials unit in Cambridge and the statistical and clinical trials units, those centres as well became involved with the design. And then, of course, we had to talk to pharmaceutical companies as we were thinking about which drugs we might want to try, having the discussions to set up collaborations with them. So that was really the starting point. And then behind all that, of course, there's an army of doctors and research nurses, and of course, a very large number of very courageous patients, who've stepped up to the plate to make this work.
Fantastic, thank you. So, Professor Wilkinson, beg your pardon. I know that researchers can't just go and start up trials whenever they think it's a good idea. So when they do have an idea for research, how do they go about getting started? You sort of mentioned some of this before, but what checks and balances are in place to make sure that the trials that we run are safe and that they're actually able to answer the research questions that we're looking to answer?
Yeah, that's an interesting question. So, it depends what type of research you want to do, really. All research requires ethical approval, even if you want to ask people some questions. But what we're talking about here is drug trials, and they are highly regulated to ensure safety for participants and to protect medical staff as well running those trials. And I have to say the UK has really led the way in the regulation of trials over the last 50 years or so. So it's got a very well-oiled machinery for doing that. So if someone comes with an idea like Francis, the first thing is to have a chat and see whether it's a sensible thing that we think we should be investing time in from the trials unit point of view. And then you've got to make a protocol to describe in some detail what you want to do and why you want to do it. Then you've got to go and get some money, because you've got to have the funding in it to enable you to do the trial. And that's, I guess, where the BRC can be very helpful. Has a lot of infrastructure, a lot of expertise in helping you to design that trial. And also the NIHR has various funding streams that you could apply for. Once you've hopefully been successful and got the money, then the hard work starts from the trials unit point of view. But there's an inordinate amount of paperwork. It is still actually largely paper, even in the modern era. That's got to be put together. Information for patients telling about the trial, much more detailed protocol, forms to collect all the data. We have to make a database, and then we go out and get the regulatory approvals. So Professor Bradley, head of R&D, would need to approve what we want to do before we do anything else. And then we go to an ethics committee, and lots of different people with expertise, lay people, statistics expertise, will ask us questions and review what we're going to do, and usually make some suggestions about how things might be modified. And then we go to the regulator for the drug trial, that will be the NIHR, and they review the safety of what you want to do. And if you're repurposing a drug, often that's easier because there's established data about the drug. It's been into people before. If you're working with a new chemical entity, then you have to produce a lot more data to convince the regulator that it's safe to do. And then you can get going. The trials unit will then monitor the progress of the trial and make sure everyone's compliant with the various regulations to make sure that it's done safely and that we have faith in the findings of the trial.
Fantastic, thank you. Professor Bradley, we are really, really fortunate in Cambridge, as you know, that we are home to some of the world's best scientists and clinicians. And yet, since the start of the pandemic, you know, there's only so many patients, even through Addenbrooke's, that we've had, which is a good thing for healthcare, of course, but for research can present some problems. You mentioned a bit about the COVID bioresource earlier, but how are we making sure that we learn as much as possible from each patient that comes through our doors, and also so that there aren't more trials than there are patients?
We, right from the very beginning, reorganized the research staff. We have a very active clinical research facility on the campus, embedded in the hospital. And we redeployed many of those staff onto the wards so that they could help with the recruitment of patients. And we hope that we offered virtually all patients the opportunity to participate in research. And indeed, at the beginning, we would have a review meeting every morning where we would try and check that we had given all patients that opportunity. They could, should they wish, join the bioresource, and that would allow them to provide us with samples. And the real advantage there was that because we are on a biomedical campus with many research facilities, and indeed some of the very high level of containment facilities that are required, we could rapidly analyze fresh samples. And indeed, many very experienced researchers changed from their roles in the lab to being couriers and running samples between the patients and the labs so that we could analyze them as quickly as possible. That gave everybody the opportunity to participate in research. As we moved on and were able to enroll people in clinical trials, they would have different entry criteria. And really, again, we hoped that we could offer all patients the opportunity to be in a clinical trial and joined up, as you've heard from Francis, with other centres so that we could share experience and make sure that the right patients got into the right trials.
Fantastic, thank you. So most clinical trials that have come across all come with their own fancy acronym, and tactic, of course, is no different. I think it suits it to a T. Dr. Hall, could you tell us, what does tactic stand for? How did you come up with the name?
Well, I have to credit my colleague in CCTU, Joseph Cherian, with this name, but the full name of the study is Multiarmed Therapeutic Study in Pre-Intensive Care Patients Admitted with COVID-19. So obviously we don't want to say that every time we want to talk about the study. And if you borrow letters from all the different words, it isn't actually the first letter of each word, but Joseph concocted devised a sort of scheme of borrowing letters from words in there to come up with tactic. And of course, that has an additional significance in that this kind of immunomodulatory approach is a distinct tactic from the dominant tactic a year ago, which was antiviral.
Of course. And in the trial at the moment, there's 2 drugs that have been selected. So how are these 2 drugs initially chosen, and what does each drug do?
Yeah, so well, I mentioned before that what we felt we were dealing with was a condition that was predominantly driven by the immune system behaving inappropriately. And so we sat down and considered candidates for of the drugs that were already out there being used to treat autoimmune conditions and conditions where the immune response is known to behave badly and cause disease. And then we looked at what was known a year ago about the mechanisms within that, under that broad umbrella of immune-mediated damage. And we could see that one set of problems seemed to be caused by chemicals in the immune system called cytokines, which were being upregulated inappropriately. And each of these chemicals or cytokines is, if you like, it's a sort of immunological WhatsApp group of its own. So each one triggers a whole pile of other messages to all of its friends and kicks off a whole range of different processes. And several of them are going haywire at the same time in COVID-19. So we thought that we should choose an agent which interfered with these WhatsApp groups. And we became aware that several other groups were thinking about using surgical strikes on individual WhatsApp groups, if you like, by using monoclonal antibodies. But we decided to take a slightly different route by choosing baricitinib, which is a medication which actually dials down, if you like, the chatter in several of these WhatsApp groups all at once, rather than taking an individual one out. And there are several drugs in baricitinib's family, but we chose baricitinib because it also, quite separately, has a bit of direct antiviral action because it reduces the uptake of the virus into cells. So we thought that was very useful to have. So that's how we chose baricitinib. And baricitinib is repurposed because it's used actually in rheumatoid arthritis. It's used at the severe end of rheumatoid arthritis. So obviously, as a rheumatologist, I was very aware of baricitinib. So ravulizumab was the second one we chose, and that targets a completely different mechanistic strand. So this inhibits a molecule in what's called the complement cascade. So the complement cascade is a group of molecules in the immune system that, if you like, acts as a tag team. So they kind of pass on the message from one to the next, activating the next person along and the next person along. So they amplify inflammation, but they also end up producing a little molecular hole punch, which really does what it says on the tin. It punches holes in cells and it kills them. Now that's a good antiviral strategy, and actually antibodies can focus this hole punching activity on infected cells. But we knew that because antibodies could focus this, and that people were getting sick when they were making antibodies to the virus, that actually this complement cascade might be important. And indeed, over the last year, a lot of additional evidence has come to the fore to show that that's the case. So that's why we chose ravulizumab, and that's also repurposed. It's used to treat a kind of anemia, which is caused by the immune system going haywire and making these hole punches and punching holes in red blood cells.
Fascinating, amazing. Professor Wilkinson, I've heard that tactic is called a platform study, which I think is the same as some other sort of well-known trials across the UK, such as Recovery. Can you tell us what a platform study is and why is it being used so often in the pandemic? So
traditionally, if you did a trial, you might pick a drug and a placebo and put them head to head and run what's called a randomized controlled trial. What a platform trial does is it allows you to add drugs or take drugs away as you go along. And it's generally considered to be a much more efficient way because you have one control group, but you can have different drugs added in as treatments or taken out if they're not proving to be effective based on some early readouts. A particular pandemic where, as I said to you before, you're compressing the paperwork and approvals that might take 6 months into weeks. It's just much more efficient to put all the effort into creating a platform, and then you add in drugs. Now, having said you can just add in drugs, there's some checks and balances around that. I won't bore you with the details, but there's some statistical methodology you've got to be aware of. The other point is you still got to put each of those new treatments past a regulator and the ethics committee as well before you can do it. So it's ideal for pandemics.
Excellent, thank you. Professor Bradley, as part of your role as Director of Research, you need to ensure that the research that we do in Cambridge complements the research that's happening across the UK and indeed internationally. So how does TACTIC and other Cambridge-led trials sort of fit in with the wider national and international effort?
As we've heard, TACTIC is a national trial, and indeed we hope we actually may be able to open sites internationally. So it does need to be coordinated, and there are a number of other centres that are working with us on TACTIC. We've heard about some of them. And then there'll be other centres that are doing different platform trials. There's one national platform trial that virtually all centres are involved in. That's the recovery trial. That's fairly unrestricted in terms of the entry criteria. Whereas in the TACTIC trial, we are looking at a select group of patients, particularly those who we think may not do so well and are perhaps more likely to respond to the drugs that are in the trial. For the other studies, we obviously collect a lot of samples. We're analysing those to learn about the disease. And in many cases, we're part of national consortia that are actually doing perhaps immunological studies looking at the response to the COVID virus. So it is all joined up and has been joined up very rapidly by this remarkable national research effort.
Fantastic, thank you. Dr. Hall, with Cambridge and so many other participating hospitals supporting TACTIC-R, can you tell us where are we up to with the TACTIC trial? Can we expect any results anytime soon?
Yeah, sure. So we have 416 patients who've been randomised to one of the arms. So that's either standard of care alone, or standard of care plus baricitinib, or standard of care plus ravulizumab. And at the moment, recruitment is obviously slow for all sorts of good reasons, like the fact that the case rate has gone right down, which I think most of us are very happy about. At the moment, so we're gathering the data and generating queries if there are any problems or issues with the data, and getting all that clean so that it can go into the statistical analysis sort of part of the study. And as Professor Wilkinson mentioned, this is designed as a platform study, and part of that design is what's called adaptive. So it allows us to look at the data and do an analysis at predetermined points. So we're at the first predetermined point where we need to look at the data. And so we anticipate having the data sort of cleaned and ready to do that analysis in May. And then based on what we see from that, and of course what's happening in the country at large, we then need to make a decision on what happens next.
Okay, very exciting. Professor Wilkinson, whatever happens next with the outcome of our interim analysis, what will the next steps be? So what happens to a treatment that appears successful in a clinical trial?
So if you have a signal suggesting the treatment's working, then in a normal situation that you might want to test that in a different trial. And certainly a regulator, before they gave a license, may want 2 different trials to prove the drug works. It's slightly different with a repurposing study, where a study on its own may be enough to grant you a license or to grant you an indication. And certainly what we've seen in the pandemic, for example, some of the drugs that Recovery has looked at, or some of the American trials, those drugs have been fast-tracked, helped because they've got a license already, and we know they're safe, and we've made them already, but they've been fast-tracked to use, and they've really got into patients incredibly quickly. So if we saw a strong signal of efficacy, that means it's working with baricitinib, for example, then I'd hope that would get fast-tracked and we'd make it available to patients rapidly. Because the whole point of clinical research is to improve outcomes for patients. And we need to therefore work very quickly, particularly in the context of a pandemic, to do that.
Absolutely. Well, fingers crossed it's good news when we come time to do those final calculations. Professor Bradley, to date, we've had hundreds of patients and healthcare staff participate in COVID studies right here in Cambridge. And certainly my team have been overwhelmed, and I know researchers right across the campus have been overwhelmed by the response of the public in their willingness to participate in these and in other studies. What would you say to our patients and anyone who's considering taking part in clinical research?
Clearly, we would encourage people to do so. If they visit our website, they can find out a little bit more about the research that we are doing and how they could get involved. I think it's important. I said at the beginning, when we started on this journey, we were very uncertain about things. There was a lot of fear, also a lot of courage. We really are tremendously grateful to everybody who has contributed to the research effort. We've got over 6,000 people now in the COVID bioresource. Many of those were recruited in hospital. There were people who were sick and who were frightened, but they were still prepared to help us with the research effort to understand and ultimately treat the disease. So it's really a very big thank you to them.
Fantastic. Well, I think that's probably about enough questions for me. So I think it's time to open up to questions from the audience. And thank you very much to all of you who have already sent some questions in. And I've been watching as we've been going along. There's some really good questions that have been put in the Q&A section. Starting with one that could be either Professor Bradley or Professor Wilkinson, and is a very good question for the many different acronyms that we started using at the beginning. Rob has asked us, what is the difference between the Cambridge Clinical Trials Unit and the Cambridge Biomedical Research Centre, and how do they work together?
Shall I start by telling you a little bit about the Cambridge Biomedical Research Centre?
That seems fair.
Which is an infrastructure across the campus funded by the National Institute for Health Research over a 5-year cycle, including funding for the bioresource. It receives just under £150 million. Part of that is the Cambridge Clinical Trials Unit, which, as we've heard, Professor Wilkinson is the director of.
Fantastic, thank you. Then we've had another person put in the chat, do you think that COVID has changed how we carry out research? Do you think that we could now speed up cancer or dementia research or research into any other subject? Professor Wilkinson, do you think you'd like that one?
Yeah, I read that. I read that in the chat as well. It's a very interesting question, and I absolutely agree with that. I think what it's shown is that we can move more quickly. I'm not sure we can work at the frenetic pace that we've done in COVID permanently, but it has not only in trials but in the health service in itself. It's tested the system, and I think what it's proven is that if we want to, we can move more quickly. And actually, we've done that safely, which is the real important thing here. So I think there will be very important lessons to be learned about making the system more efficient, because if we can get the drugs tested more quickly, but robustly and safely, we can get those treatments into patients. And I really feel this is something that the UK needs to capitalise on after this. It's led the way with innovative treatments for COVID, and it should be able to do the same for other diseases, as was mentioned.
Thank you. Actually, that leads quite nicely into the next question, which says, if you could go back in time, what's one thing that you would have done differently to handle COVID, or that you would do first in another pandemic? Dr. Hall or Professor Wilkinson, either or.
So I'll just unmute myself. I can start off with that. So I think Professor Wilkinson has mentioned how relatively quickly things got done. I think it would be really hard to improve on that, because it's perhaps not obvious, but many people work through evenings and weekends for many weeks to get this off the ground. And of course, the landscape has changed very much over the years. So looking back, we might have changed things, but it would have been impossible to quite see which direction things were going to go in at the outset. I think one thing we have done that we should have done more of, and we would want to do more of, is dock together every step of the process with basic science research. And when we have done that, we realised that was a priority. And of course, it was a huge rush to get all of this up and running. But that is so important that as we gather the clinical data, we also gather as much data as possible to not only see what works, but to understand what's going on and why it works. I think that relates to some other questions that are coming up later. So I guess that's one thing that we would do differently. And I think another thing that we've done well, but perhaps could do more of, is brokering cross-centre collaborations. So you pull together the the strengths of not just the researchers in one centre, but actually across different centres, as we've done with King's College and Guy's Thomas's. We have had input from some other centres as well, for example, Glasgow. So I think that's been powerful, and that's something we could do more of.
Fantastic. I've got one that's about the immunology of COVID-19 that says, why do some people get sick from COVID and others don't? Dr. Hall, I feel like
shall I take that? Yeah, that is fascinating, and I wish I could give you a full answer. There are a lot of people working on that, including people who are working with bioresource material in Cambridge, as Professor Bradley has mentioned. I mean, it's clear that there are very big differences between people. We know that the immune system is highly variable between individuals, and that's actually generally a great strength, because if we were all the same, a pathogen would be able to find our Achilles heel and exploit it and wipe us all out. The fact that we're not all the same in the way our immune response behaves actually is a great strength, for us as a group. But unfortunately, for any given scenario, some people's immune systems perform better and some worse. And we are starting to understand the basis for some of those differences, but we're way off others. So underneath a lot of this is something called polymorphism. So in genes, like hair color, for example, some people have got fair hair, some people have got black hair, some people have got ginger hair. Well, with the immune system, there are a lot more variants than just those. In many genes controlling immune processes, there are multiple different variants. And this gives rise to different behaviors when the immune system's challenged in a particular way. So some of the cytokines, chemicals I mentioned, these WhatsApp groups, some of the WhatsApp groups, if you like, in some people, have a few followers, and some of the WhatsApp groups have large numbers of participants. And so people vary enormously into what happens when that chemical gets released. Some people have a little response, some have a moderate response, some have a big response. And one of the key chemicals is a form of interferon. So many of us will have heard of interferons, and we're aware that they tend to go up when we have viral infections, and that's part of our rapid response system to viruses. Well, it turns out some people tend to put a little bit out and some people put a lot out. And for example, people who put a lot of interferon out early on seem to do better with COVID. They tend to get SARS-CoV-2 infection, tend to sort of resolve it more like a sort of standard viral infection. People who don't put out so much at the beginning might actually not feel so sick early on, but then they bring in other immune or parts of the immune army, and some of those sometimes go wrong, and the chance of them going wrong again will depend on all sorts of genetic settings. So there's genetics, but then there are also environmental things. So we know that age, but also whether you're thin or whether you're a little bit overweight or a lot overweight, all of those things sort of change the way the immune system works, change the way that you handle the virus. So we've learned a lot, but we've got an enormous amount more to learn about that.
Excellent, thank you. We've got just a few minutes left, so I think we've probably got time for perhaps 2 more questions. Someone in the audience has asked, in your opinion, will COVID be here to stay? And is it something that we'll just have to live with? Do you think we'll be having to have medicines ready at all times, or will it just sort of go away on its own? I'm not sure who that question's sort of best at. I'll leave it to, yeah.
I can take that again. I'm afraid I think it is here to stay for the foreseeable future. Hopefully rolling out the vaccines will increase the protective immunity in the population, but of course we've got to think globally, not just nationally, to do that. And the problem is, the longer it takes us to roll that out, the more the virus carries on replicating, the more chances it's got to mutate and get itself into a configuration that can evade the antibodies that the vaccine has raised. So I know the vaccine teams are working on making new versions of the vaccine to cope with this, but it's an arms race. The virus is evolving and we're evolving, to catch up with it. So I think we've got it for some time to come, probably permanently. We need to know not only how to immunize people against it, but we also need to know how to treat it. So running clinical trials to figure out how to treat COVID isn't just something that's useful until we have a vaccine. We're always going to have a group of people who get COVID because they haven't responded to the vaccine, or because there's a new variant of COVID. And so, understanding how to deal with that, and indeed what's going on in the immune system is really important for that reason.
Fantastic, thank you. I think for one last question, someone in the audience has asked whether or not we are considering research into long COVID now that we're having so many survivors. Is there any research being done in that area? And Professor Bradley, I think that's probably a good question for you to finish on, perhaps.
And the answer is yes, in two ways. Firstly, anybody enrolled in the bioresource, we're following over time. And so we're just starting to see people a year after they first presented. And we also set up a clinic that allows people to be referred in from the community who may not have been to hospital. Some of them may not have been positive in terms of diagnostic tests for COVID, and we can do more detailed analysis, looking not just at antibody responses, but cellular, and in particular, what we call T cell responses to the virus. So yes, we are. Obviously, that inevitably came after the initial research, but it's something that we're really starting to build.
And hopefully that will be along with the focus of the next phase of our research. Well, thank you very much to everyone who's joined us tonight. I'm afraid that's about all we've got time for. And we certainly hope that you found the discussion interesting. Could you tell us whether or not you feel like you have learned anything from what we have shared this evening? So I've got just put a poll up where hopefully you can tell us if you feel like you've learned something. It looks as though most of you have learnt something this evening, and it looks like we've all done a very good job. So thank you very much to my panelists for joining us tonight. We really appreciate the time that you've given us and your extremely thoughtful answers. So thank you very much for your time. I would also very much like to thank the members of my team, the members of the NIHR Cambridge BRC PPI and comms team, who were my co-pilots for this evening, and without whom I would never have been able to set up this event. So thank you very much to them. They've also been working very, very hard on an animation that we have about the Tactic R trial, where you get to meet Baz and Rav, the two treatments that we meet in the trial. And we very much invite and encourage you to check out that animation on YouTube and to download the associated quiz and colouring in sheet if you've got some kids who might like to learn a little bit more. We also hope that when you leave us tonight, you'll take a few minutes just to fill in our feedback survey and help us to learn what we need to know if we run a similar event next year. So we do hope you take a few minutes to do that. If you are interested in research, we also invite you to consider becoming involved in research as a member of the public. So here in Cambridge, you are very, very welcome to join our patient and public involvement panel, which is a group of people from in and around Cambridgeshire who are interested in research and who provide feedback, ideas and life experience to support research into a wide range of conditions. We don't expect you to have any experience of healthcare to be able to join us, and we really are looking for people from a whole as wide a range of backgrounds as possible. So if that does sound interesting to you, then we hope that you will use some information on the slide that we're about to put up and that you'll consider joining us. And I know Georgina's going to put some information about that in the chat. So thank you all very much again for coming, and we wish you a very safe and pleasant long weekend. So good evening and good night.